Tissue culture. It is a term used for the process of growing cells artificially in the laboratory. Plant tissue culture is a technique of growing plant cells, tissues, organs in an artificially prepared nutrient medium under aseptic conditions. Types of tissue culture. Callus culture. Callus culture may be defined as production and maintenance of an unorganized mass of proliferative cell from isolated plant cells, tissue, organ by growing them on artificial nutrient media in glass vials under controlled aseptic conditions. Anther culture. It is the androgenesis is in the vitro development of haploid plants originating from potent pollen grains through a series of cell divisions and differentiation. The next type is the pollen culture. Pollen culture is the in vitro technique by which the pollen grains, preferably at the microspore stages, are squeezed from the intact anther and then cultured on nutrient medium, where the microspores without producing male gametes. Embryo culture. Embryo culture may be defined as aseptic isolation of embryo of different developmental stages from bulk of maternal tissue of mature seed or capsule, and in vitro culture under aseptic and controlled physical conditions in glass vials containing nutrient semi-solid or liquid medium to grow directly into plantlets. Coming to the anther culture. Androgenesis is the process in which embryo develop without genetic contribution from oocytes. In androgenesis, the male gametophyte, that is microspore or immature pollen, produce haploid plants. The basic principle is to stop the development of pollen cell into a gamete and force it to develop into a haploid plant. There are two approaches in androgenesis. The selected flower buds of young plants are surface sterilized and anthers removed along with their filament. Then the anthers are excised under aseptic condition and crushed in 1% acetocarmine to test the stages of pollen development. If there are at correct stage, each anther is gently separated from the filament and intact anthers are inoculated on a nutrient medium. Injured anthers should not be used in cultures as they result in callousing of anther wall tissue. The anther cultures are maintained in alternating periods of light and darkness at 28 degree centigrade. As the anthers proliferate, they produce callus which later form an embryo and then a haploid plant. Pollen culture. Haploid plants can be produced from immature pollen or microspore. The pollen can be extracted by pressing and squeezing the anthers with a glass rod against the sides of a beaker. The pollen suspension is filtered to remove the anther tissue debris. Viable and large pollen are concentrated by filtration, washed and collected. These pollen are cultured on a solid or a liquid medium. The callus or the embryo formed is transferred to a suitable medium to finally produce a haploid plant and then a diploid plant on colchicine treatment. Comparison between the anther and the pollen cultures. Anther culture is very easy, quick and practicable. Anther walls act as a conditioning factor and promote culture growth. Therefore, anther cultures are reasonably efficient for haploid production. The major limitation is that the plants not only germinate from pollen but also from other parts of anther. This results in the population of plants at different ploidy level. They can be diploids or can be aneuploids also. The disadvantages associated with anther culture can be overcome by pollen culture. Many workers prefer pollen culture even though the degree of success is low, as it offers the following advantages. The embryo culture. Embryo culture may be defined as aseptic isolation of embryo from the bulk of maternal tissue of mature seed or capsule, and in vitro culture under aseptic and controlled physical condition in glass vials containing nutrients, semi-solid or liquid medium to grow them directly into plantlets. Culture of embryo may be divided into the following categories. Culture of media and intact seed embryo. The aim of this study is to analyze the various parameters of embryonic growth and the metabolic and the biochemical aspects of dormancy and germination. Second one is culture of surgically dissected embryo. The mature seed embryo can be dissected surgically into a number of segments. Such embryo segments are cultured to analyze the relationship of different parts of embryo to its final form in culture. The third one is culture of immature embryos or proembryos. The term proembryo means early developmental stages of embryo that precede cotyledon initiation. Globular and heart-shaped stages of embryo are approximately called as proembryo. The objective of such culture is to understand the control of differentiation and the nutritional requirement of such progressively developing embryos. Fourth one, culture of intact seed containing undifferentiated embryo. Each fruit of an orchid plant develops several thousands of tiny seeds which contain morphologically undifferentiated embryos. There is even no storage tissue in the seeds, and the seed coat is reduced to a membranous structure. For this reason, the entire seed of orchid containing undifferentiated embryo is cultured and treated as embryo culture. The fifth one is, besides the zygotic embryo produced from egg cell, some additional embryos are produced from nuclear tissue in polyembryonic seeds like lemon and oranges. Such additional abortive embryo sac can be exploited in culture for clonal propagation. Sixth one, culture of inviable or abortive embryos. In many interspecific or intergeneric breeding experiments, sometimes inviable or abortive embryos may develop due to unsuccessful crosses. As a result, the non-viable seeds do not germinate normally. But it is now possible to raise a hybrid plant by culturing the inviable embryos in vitro. Coming to the embryo culture steps. The surface sterilization of embryo is not required because it is already packed in the sterile environment of fruit or the seed. However, after the collection of the fruit or the seed, it is required to send them under surface sterilization. After this, you can safely excise the embryo under a sterile environment for culturing process. If a tough cover is present over the seeds, then surface sterilize those seeds with its seed coat and soak them in sterile water under aseptic conditions. This will loosen the outer covering of the seeds and make the incision of embryo easier than before. However, when you are culturing very small seeds, it is recommended to excise the embryo under a microscope. This will help to protect the embryo from damage that can be caused during excision. In some cases, like orchids, whole seeds are cultured because of extremely small size of seeds. Application of embryo culture. Sterile seeds can be produced using embryo culture techniques. Micropropagation of hard species is easier using embryo culture technique. Embryo culture is used for production of haploids in case of Hordeum vulgare and Hordeum bulbosum. Embryo culture is also used to propagate parasitic plants that require a suitable host to complete their life cycle. These plants are called obligatory parasites. These plants die if they do not find a suitable host to fulfill their requirement. Thank you.